So, my name is Alena. I am in England. That's why I'm speaking in English. I am at the Sanger Institute, and I am here today to talk to you about DNA. So, just to have an idea, you can either use the chat or mainly tell your teacher and to see who knows about DNA. How many of you have heard of DNA?
Guys, you can answer.
I have.
Oh, wow. Very good. So that's a very good start. So
I've heard that DNA is it kind of makes how you look like, how you work, and other stuff.
And where is it, this DNA? Who's got it?
In our cells.
It's in our cells. Yeah. Wow. Well, you've jumped ahead a few slides, but let's take it one by one, shall we? So here, if I show you these, who's got DNA? Do these have DNA?
Yes.
Yep.
Lions?
Animals and humans.
Yes.
And what about the plants?
Yeah, plants got two.
Yes, I feel plants too.
Yes.
So, and what about this picture here? So this is a microscope picture of very tiny things, microbes. Do you think microbes have DNA?
Yeah.
Yes.
I think it.
Yes.
Very good. And what about that the rock that the penguins are standing on?
Penguins, this is.
No.
I think
No.
it has DNA.
So the conclusion is all living things have DNA. So where is it? Well, we, represented by this man here, are made up of trillions of cells. And every single one of those cells has a compartment inside that is called the nucleus. And in the nucleus is where the DNA is. And because there is so much of it, it's all scrunched up into structures called chromosomes so that it can fit. It's like if you take a sheet of paper and you scrunch it up really a lot so that it gets very small. That's more or less what happens. So if you then get the chromosome and you unscrunch it, then you end up with the structure, this structure here, of the molecule of DNA. Does anyone know what is it called?
I know.
Go ahead. It's called, maybe you don't know in English. In English, it's called the double helix because it is a helix of two strands intertwined with each other. So what does it do, the DNA?
Question: why is the chromosome an X? Why not a T? Why not like a Y or anything else?
That was very loud and it resonates very much. So I didn't understand the question.
Why are chromosomes X's? Why not A's? Why not any other thing?
Because actually the structure of the chromosome is very important for its function. And the bits out here on the outside of the X's are called telomeres. And the genes that are there are different and have a different functions from the genes that are in the middle, which is called the centromere. And also the way it replicates is different. So the shape of the chromosomes has to do with their function. Coming back to the question I asked before, so what does the DNA do?
The DNA has the information that that is in our in us.
DNA save information about people.
That is very good. The DNA makes us the way we are. So if we have blue eyes, if we have dark hair, if we're tall, but as you correctly said, the DNA actually doesn't do anything. It contains the information. So it is, in fact, an instruction manual. It's an instruction manual that tells the cell what to do and when. And this is very important because this is the only way that cells can function and that big organisms like us can actually function and exist properly. So let's have a closer look. You have here the double helix. And if we unwind it, we find this structure. This is a cartoon, obviously. And the double helix is formed by two strands. You have them here, and you can see them here as well, of the building blocks of the DNA that are called nucleotides or bases. And here they are. There are four of them: thymine, cytosine, guanine, and adenine, or TCGA for short. And in each strand, they are sort of holding hands together, making one strand. And then the two strands stick together by interactions between the nucleotides. So they are paired together, but in a very precise way. So thymine is pairs always with adenine. Just a minute, I need to get the dog out. Right, go. Sorry, that's what happens when you work from home. So the nucleotides interact in a very specific way. Thymine always interacts with adenine, and cytosine always interacts with guanine. And you can see it here, where you see adenine and thymine and guanine and cytosine always pairing together. So when we look at the DNA, all of the DNA of an organism, be it an animal or us or a microbe, is called the genome. That's the whole manual. But within that manual, and taking the analogy, you know that a manual is full of text and there's a lot of text, but within that text, there are very short, specific, and very concise sentences that give the actual instructions. And these we call genes. So genes are fragments of the DNA that contain the information necessary to make a protein. And it's the protein that will make what the cell needs to do. So it's the protein that will perform the functions of the cell. And if you look at your hands. Look at your hands. What are your hands made of?
It's made of skin.
Yes?
Cells.
Repeat what you asked just now.
They're made of cells.
Mm-hmm.
Skin.
Yeah, very good, very good. So if we look at
Veins and arteries.
yes, that's exactly it. So if you look at your hands are actually very complex. And your hands are made of skin and muscle and bone and cartilage and veins and nails and blood. And all of these tissues are made of cells that are very different from each other. And that's because the cells of the skin have the proteins that they need to perform their function. And those proteins are very different from the proteins that you find in muscle cells, because the proteins of muscle cells perform the functions of the muscle. And so all of these, the sets of proteins that are in each different cell that's because the genes for these proteins are active in these cells. And that's why the cells are so different, because even though every cell of the body has the same genome, the genes that are actually active and making proteins are very different from one to another. And again, that is very important because that's how we have different organs that work differently. So we said that we are made of trillions of cells. So when a cell divides to make 2 new cells, what happens to the instruction manual?
They are to
It's DNA and put it to like
It also divides.
You can't have a cell that then takes the manual and the other cell doesn't have anything, because then that cell wouldn't know what to do.
Well, that's what I'm saying. The chromosome divides.
So every cell has to have the manual, and so the manual has to be copied. Yeah. So how does that happen? In a nutshell, what happens is that the strands of the DNA separate, and then the gaps are filled with the corresponding bases. Because we said, yes, because we said the bases pair in a very specific way. Yeah. So let's have a look at it in a bit more detail, right? So we start with a double-stranded DNA. This is a cartoonish way of representing it, but you have the 2 strands here. So the first step is to separate the strands. Yeah, and that's a protein that does that called helicase. You can see the DNA here. Yeah, and then once the 2 strands are separated, then comes the DNA polymerase and puts the corresponding base on each of the strands and makes the second strand, and you end up having 2 full copies of a double-stranded DNA. That's how it gets copied. So how about we see whether you got it? So we have here a stranded DNA, and you are the polymerase. So if you are the polymerase and you're complementing the strand, what base are you going to put here?
C.
C.
G.
And then the next one?
C.
C.
And the next one?
C.
And the next one?
C.
A.
C.
And then?
A.
T. T.
T.
T. T. T.
Got it! Bravo! So that's how DNA replicates. There we go. So of course DNA is, and you know, if we were together physically, we would have a game to play this out, but we can't do that. So let's talk about something else. The DNA, therefore, we've agreed that it's very important. It contains all the information that we need for our cells to function and for us to be the way we are. But What else can we do with it? What do we do with the DNA? Well,
Copy.
we sequence it. So this is the sequencing lab in the Sanger Institute.
What does sequence mean?
I don't know.
I'll show you in a minute. So I have here an example of a gene called luciferase. And it's a really funny gene because it is the gene in the firefly. You know what a firefly is? When you're out in the night, in the trees, you can see these little insects that glow.
They're beautiful.
And they're very pretty. So the protein that makes them
A lot of words.
the protein that makes them glow is called luciferase. And this is the sequence of the luciferase gene. So you can see that it is just a string of A, T's, G's, and C's. And that is what a sequence is. It means the order of the bases in the DNA, in the gene that makes that particular protein. So
Lots of letters in the right order.
and they have to be in the right order because otherwise you would have something else. So I could read all of this, but I'm not going to. But what can we do? I mean, why is it so practical to have the sequence and to know the order of the nucleotides in the gene? Is because we can then take it and make luciferase in a petri dish or in a tube in the lab. And we can also, have you heard of gene editing? It's
What?
gene editing. Gene editing is a novel, it's relatively new technology.
Gene modifying? General?
Gene editing. It's like editing a text. You change a letter here and there and make it different. So
People very speak fast.
am I going too fast?
Yes.
Sorry. So when we have the sequence or the order of the nucleotides in a gene, we can then take that sequence and use it in the lab. So we can make the protein, luciferase in this case, out of that sequence. And that's very useful because we can then study how the protein works, which are the parts of the protein that have different functions and how does it all work together. We can also change some of these letters and then make that protein which will be different and see whether it still works or not. And that gives us clues as to whether those changes have changed the function of the protein or not. And if you extrapolate that into a wider context, you can, and this is being done quite a lot nowadays, you can sequence, I have the order of the nucleotides of the whole genome of a cell. And if you do that with a healthy cell and with a diseased cell, and then you compare the two sequences, you might be able to find the changes in the diseased cell that might give you a clue as to what is wrong with that cell and why is the disease. And this is very useful for medicine because it helps us understand how disease happen and therefore find ways to cure it or treat it. And I'll give you an example with this little story. So you have here a little sequence, DGTCAAC, of a normal piece of the genome. And then you have the same sequence, but in a cell that was exposed to too much sun. You know, when we all go to the beach. And
Я слушаю. Иди там.
and then you have another copy of the same sequence, but that comes from a cell of someone who smokes a lot. Can you see any differences?
Is this the son Dana?
Sorry?
Is this the son Dana?
It's I don't understand what you're saying.
DNA.
Yes. Oh,
DNA, not DNA.
DNA. Yes. So this is the sequence of the DNA in one cell that is healthy, and then you have a cell that has been exposed to the sun very much.
If the skin was added to the if too many sun T added, and if smoking A added.
Yes. So, my computer is playing up. So, yes. So there is a change here, and a change here and here, in this case, and then you have changes here as well that are due to the tobacco. And all of these changes will mean there will be protein that won't be functioning properly, and so therefore the cell might develop a disease. And that's what the sequence of the genome or of particular genes is used for. And it's very, very useful for medicine, and it's also useful to try and find what are the genes that are causing a disease if they are damaged. And I think with that, I won't complicate things anymore. I thank you for your attention.